Hello, my name is Margaret Gleason. I am a graduate of St. Mary's College.
And my name is Valerie Eddington. I am a senior biology major at St. Mary's College.
This research is the result of further analysis on my undergraduate research at St. Mary's College. Bats and dolphins are best known for echolocation, but additional animals use ultrasonic signals to sense their environment, including swiftlets and shrews. What these animals all have in common are short-duration ultrasonic signals that are made often with head scanning behavior.
Focusing on shrews, who have been documented producing ultrasonic clicks, a recent study proposed that these clicks could be a product of locomotion. In our study, we wanted to explore this further and assess whether shrews produce ultrasonic clicks in the absence of any substrate and in the absence of locomotion when placed in a novel environment. We also wanted to examine the differences in signal parameters between individuals to see if the signals are consistent. This can indicate if the signals are a behavior of the species rather than a behavior specific to the individual.
We caught all of our shrews in the St. Mary's College Nature Trail. The image on the left of this slide shows one of two fields that are located along the forest edge where we set the traps. In the fall season, 1 hour before sunset, we set up 16 sites, each with 2 Sherman live traps, one with a mixture of oats and Critter Crunch, and the other with dried mealworms. We checked the traps about an hour after sunrise. If a shrew was caught, it was transferred to a transport container. If a non-shrew was caught, it was carefully released back into the wild. Shrews were transferred to a care facility complete with bedding, shelter, and food and water. They were not held any longer than 72 hours and were released back into the wild after we finished with recording sessions.
Our experimental setup, pictured here to the right, consisted of 2 infrared lights on tripods, 2 infrared cameras, one attached to the bin and one in the bin, an ultrasonic microphone, and an experimental chamber. The experimental chamber was lined with felt to help prevent click echoes in the acoustic recordings. The bottom of the bin was lined with graph paper in order to determine how far the shrew was from the microphone when the clicks were made. Prior to recording sessions, shrews were deprived of food for 1 hour in order to elicit exploratory behavior. The equipment was turned on and the acoustic and video recordings were synced before the shrew was placed in the bin. Once the shrew was placed in the experimental chamber, the overhead lights were turned off and all people present exited the room. The shrew was then recorded for 10 minutes. We also conducted a series of control recordings to make sure that the sounds produced were in fact emitted from the shrew.
Click trains, defined by a series of more than 5 consecutive clicks, were extracted and measured. We counted the number of clicks and measured the intercall interval between each call within the train in the program Audacity. We also noted the corresponding behavior during call emission in the video. We compared these parameters between each of the individuals using a Kruskal-Wallis rank sum test.
To evaluate the highest quality signals and the ones most likely to be pointed directly at the microphone, signals with a signal-to-noise ratio above 15 decibels were extracted for further analysis. Signals and their parameters were extracted using Raven sound analysis software. Signals were categorized as either broadband, narrowband, or low frequency based on the acoustic parameters. Here we have examples of the spectrograms of our 3 signal types. Broadband signals were the most common signal type by far, and there were only a handful of narrowband and low frequency signals. Because of this, we further considered only the broadband signals and compared the differences among individuals. A Kruskal-Wallis rank sum test was performed for both the bandwidths and peak frequencies with a Mann-Whitney U post hoc test.
In Figure A at the top, we show the different behaviors exhibited during click trains for each individual, as well as collectively. S is scanning behavior, when the shrew is just moving its head up and down or side to side. M is moving behavior, when the shrew is walking around the chamber but not moving its head and scanning behavior. SM is scanning and moving behaviors exhibited at the same time, and ML represents when the shrew appeared motionless. As shown for all 3 shrews, the main behavior exhibited during click trains was scanning while moving in the environment. Shrews also exhibited ultrasonic vocalizations while both in motion and when it appeared motionless. In figures B and C, we show violin plots for the number of clicks per train and the intercall interval. These did not vary greatly, however we see significant differences in the pairwise comparisons except between individuals 2 and 3.
As you can see in Figure A, we found a significant difference in click bandwidths among the 4 individuals, with Mann-Whitney U test indicating that individual 1 had significantly higher bandwidths than individuals 2 and 3. As you can see in Figure B, There is no significant difference in the peak frequencies between the 4 individuals. Unfortunately, due to the limitations within our software, we were unable to statistically assess the signal duration, but overall, clicks were either 60 or 120 microseconds in duration.
In this study, we found that shrews emit ultrasonic noises while they are both moving and not moving. The results are also consistent with previous research that shrews produce ultrasonic noise in general. Though the function of these calls is still undeterminable without future research, there are trends that suggest that shrews may use these for echo orientation. The main behavior exhibited during call emissions was scanning and scanning while moving, which is consistent with other echolocating behaviors seen in bats and dolphins. There were significant differences in pairwise comparisons for some of the call parameters except between individuals 2 and 3. We are unsure why this inconsistency occurs and would need to record more shrews to see if the trend persists. If the calls were behavioral and used for echo orientation, we would expect future trends to show that there is little or no variation in the call parameters between individuals. What we found really interesting was the broadband signals shrews made are similar to the click echolocation signals that dolphins and cave swiftlets make. As shown in the figure on the top right of the slide, both cave swiftlets and dolphins exhibit a broadband click that they use as an echolocation signal. This suggests that shrews may use these calls for echo orientation. However, again, we need to investigate more to support this hypothesis.
Future experiments will investigate how shrews modify acoustic behavior in different cluttered environments.
This research was funded by the Helen Kuhn Carey Fund and the Tri Beta Research Grant. We would also like to thank the professors and students who assisted us on this project.
Thank you for your time, and we look forward to answering your questions.